this afternoon section dedicated to the project, to Humus project, Holim project, and Legumed project. We have already introduced the topic of our research, so thank you to be here, thank you for your attention, for your participation. And we can start directly in order to remain in time. So let's go, Marco, please open this section with the first speaker.
Okay, so we have many speakers, and some of the talk are two people that are talking together. So I ask everybody to stay on time, because of course at the end of the meeting this room will be closed and we have to leave it. So I will make a signal when 5 minutes are remaining before the end, so the speaker can adjust the talk with that. And I ask also the speaker to leave a little time for questions. Okay, let's start. The first speaker is Michel Zakai from Israel. And the talk concerns a project called Legume in Climate Change Scenario.
Good afternoon, and thank you for inviting me. I will start with a few words of hope that more peace will come to our region, the whole of our region. And I'm happy to be here. Nevertheless, I am sorry I will have to leave earlier than the end of the meeting. So I'm coming from the Department of Life Science in Ben Gurion University in the south of Israel. And I will talk about a collaborative project we have with the University of Firenze. And is it this one? Yes. Thank you. And these are the teams. So this project is financed by the Ministry of Science in both Italy and Israel. And these are our teams. So I present myself. I am I'm a plant biologist, and I'm specialized in flowering and in flowers, in flowering in general. My colleagues in Israel are David Bonfil, Dudi Bar-Tzvi, and the team Stav Biran and Rachel from Shami from the university. And the Italian team is led by Professor Federico Martinelli, who you know, with Professor Mangoni, Gonelli, and Professor Papini, who are from the Firenze University. So chickpea is mainly produced in, do you have, the pointer? Mainly produced in India, but there is also some production in our area, with Turkey being the most important in the chickpea production. And it's growing with time. Okay, so a little bit about the plant chickpea and the environmental constraints. So this is the development of the, I'm so sorry. This is the development of the chickpea plant. Thank you. One before is this. Yes, okay, so thanks. This is the development of the chickpea plant. So you can see there is two main developmental stages. So the first one is the vegetative before flowering, and then there is flower induction, and we have the reproductive stage, and of course, after we have the flowers, we are having the pod filling, and this can continue a long time. But the environmental constraint, like heat and lack of water, will lead to a stop of the pod filling, and this is called the terminal drought. So the plants, you can play a little bit with the genetics of the phenology when plants will flower. So you can have a scenario where the plants will be late flowering, so you will have a lot of time to develop the vegetative part, make a lot of leaves, after that to feed the pods, or you can have an early flowering type, which will have less time for vegetative development, but this type will, in fact, it will escape better the terminal drought. And so many cultivars which are cultivated now, they are more early flowering because of this reason. So in our project, we used a near isogenic line produced by Professor Shahal Abu from the Hebrew University and Dr. David Bonfield from the Volcani Center. And these lines were produced to check, in fact, the difference in phenology between late and early flowering types, and these types were also differing from the morphology of the leaf. So, so they made crosses between an early flowering with this type of leaf and a late flowering with this type of leaf, and continue to select for the different types of leaves and early and late flowering with the near isogenic lines. And there are many lines we have now, and we use them, which offer diversity. And we know if they are late or early flowering, and we can correlate this parameter to other important parameters. Okay, so the goal of our common project, modern cultivars, as I said, they are early flowering usually, and like always, it's a common problem. The genetic basis is quite narrow. So the project here was to use genetic diversity to find parameter genes, factors, molecular, physiological factors. Closer. Okay. So to find factors, genetic, epigenetic, molecular, or other factors that can be used in breeding to make more resistant or more tolerant cultivars, and to study all these traits using diversity, genetic diversity. And this genetic diversity, in our case, in Israel, we use the near isogenic lines, and in Italy, they use local population, different old cultivars. But I will talk today about the near isogenic lines. Okay, how does it work, the collaboration between us? So during the first year, we made field and greenhouses, sorry, experiments to check the effect of drought, or more, it's more specific irrigation, different regime of irrigation, temperature and heat stress. And then we send sample to Italy for analysis of transcriptome, and further later it will be epigenetic analysis as well. We also checked some parameters of nutrition, but I will not talk about that. During the second year is the 2-year program. So second year we selected interesting lines and we repeated the experiments. And we are about to check the expression of selected genes to try to check their relation with the parameters of tolerance to drought or heat. And of course, the part of dissemination, and that's why I'm here today. So we start with the field experiment, and it is located in the south of Israel here, and that's it. That is how it looks from above. David Bonfield, he loves to fly drones and take beautiful pictures. So you can see the different, there were 2 sowing dates, and so you can see the difference in the plants, and this is my team taking samples from time to time in the field. So the design, we want diversity, so we have 10 pairs of near isogenic lines, 5 cultivars, 2 sowing dates. The sowing dates allow us to check the effect of temperature. The sowing date can be early or later. When it's later, it's like in Italy, the weather. When it's later, so it's more hot. So the pod filling is happening when it's hot, warmer. And in each sowing date, we have several 2 irrigation regimes, and we took plant tissue sampling for further analysis. So this is how it looked. Don't worry too much about all the lines, but it's just to tell you that everything was very monitored, and we had the monitoring of exactly how much water we have from irrigation, from the sky. We know when the plants started to flower and to start their filling. And so I will show you several selected results. First of all, you can see here the colors, the green. These are you remember the near isogenic line, and they differ by their flowering time and by the shape of their leaf. So the green are the late flowering, and the brown is the early flowering. And you can see here that in each pair, you can see really the difference in flowering time, but it's less pronounced during the pod filling time, the pod time. How about the yield? So here you can see the response of all the lines to the different water regimes. And what is interesting here is that we get a lot of variation among the lines. That's what we wanted. Everything was done, of course, in replicates, so we can make statistics, and we can choose in these lines the interesting ones that reacted differently to the irrigation regime and to the temperature. And these are 3 lines that we selected. One is a cultivar. It's a reference cultivar, and these 2 are 2 different lines, and we have the early and the late lines of each from each one. So we can see here that these 2 lines here, the cultivar and the line 224, they're less affected by the drought, as you can see here from this line. During the second sowing, so the second sowing is during a more warm temperature, and this one is more affected by the shortage of water. But on the other hand, this line could reach higher yields when you water it. So this is just an example to show you that this variation among the lines can give us a lot of information to pick up factors, to pick up traits that are important for different breeding strategies. So in this way, it was a good result. So another experiment we did was to check the effect of heat stress. So this was short heat stress, and this was done in the greenhouse. The design was, again, all these lines. and cultivars. We had two chambers, identical chambers in a greenhouse, and in one of them we keep the control temperature all the time, 28 day and 18 at night. But in the other room, we made a heat stress. We stressed them for four days at very high temperature during pod filling. And the result, as you can see here, also we have a lot of variation among the lines. Here also we selected for interesting lines which had a different behavior towards as a response to the heat stress. And these are the same lines. I talk about them, I talk about in the field experiment. They also show different in their response to heat stress, as you can see here. These lines, this line, the late and the early flowering and the late flowering, they were not affected by the heat, while these lines were much more affected by the heat stress. I am aware there is a lot of variation in this statistic, but it is, you will see later that this response is backed up by other results we received. Okay, so from these experiments, also from the field and from the greenhouse, I mean from the drought and from the heat stress, we took up some samples, collected samples during pod filling, and we took the leaf here. The sample was the leaf underneath the pod, and we took these samples before the heat stress, during the heat stress, and this was a very bad experience. It was so hot in the greenhouse, we did that very fast. And after the heat stress, when the plants recover, and then we extracted RNA, sent them to Federico in here, in Firenze, to make the libraries. And we received not long ago the first results of this experiment, so that's what I will show you now, just from above, several results. So we are talking about differential expression of genes during the stress as opposed to before the stress. So what happened during stress? And as a reminder, in this tree line, we checked the tree lines, and as a remind you, these two lines are were less tolerant to heat, and this was more tolerant to heat. And here you can see the the specific genes which are differentially expressed in this situation. 5 minutes, okay. In this situation, and we can see that the more tolerant lines have much less genes that were changed during the heat stress. In the enrichment analysis, I'm sure it's complicated to see, but in several of the processes which were the genes belong to, we can see here the gray part belong to the line which is more tolerant, and the lines which are less tolerant to the heat, their genes are differentially expressed much more than in the tolerant line. And this was also a remark when you go deeper into the different metabolic processes and of it's very clear here when you look at the genes which are related to the GA and ABA related to stress, you can see a clear difference between the lines in the differential expression of the genes between the lines, which are more or less tolerant to the heat stress. So these are interesting results, and we are going to dig into them further and to make more correlations and check other parameters. So to summarize, we found indeed variation for drought, temperature, and heat stress in the lines we checked. And we have now indication about molecular factors that can be linked to this different response between the lines and the difference between the tolerant and less tolerant lines. And this is interesting and can be further used and applied in other breeding. So I'd like to thank the Israeli team, all the team I mentioned before, and also Professor Shachalabu from the Hebrew University, the Italian team. And thank you for your attention.
Thank you. Thank you, Michelle, to be on time. So we have time if there's a question.
No question?
No.
Please.
There is a very high variability in field results. This variability is due to the feature of the cultivar, or in your opinion?
These are what you saw are the means for each line. So it's not environmental, it's really the genetic variability. It's what we were looking for.
Thank you.
Welcome.
Okay, so no more questions. We can move. Thank you again.
Thank you.
To the next presentation. Here we will have 4 presentations of the project all in. And the first one is by Agnese Bellabarba and Camilla, and it's work package 1, selection of Rhizobia strain to boost alfalfa production in drought condition, genetic determinants and phenotypic analysis.
Okay, so,
please.
good evening, everyone. I am Agnese Bellabarba, postdoc at the University of Florence with the group of the Professor Carlo Viti. Today, we are going to talk to you about our results that we obtained during the All-in project, and also thank to the help of our partners from University of Oran, University of Meknes, and from INRA. As Carlo introduced you before, the All-in project were divided in 3 work packages. With Francesca and Camilla, we are going to talk to you about our activities of the first package that was dedicated to the elite inoculants to improve alfalfa yield in harsh condition. And with Camilla, specifically, we are going to show you our results of the first 2 tasks. The first task foresee the exploration of the competitive abilities of Rhizobian strain during the first steps of the symbiotic process, because, as Carlos said this morning, in nature, more Rhizobia strains compete for the plant infection, creating the so-called competition problem in field. So the competition abilities are featured that has to be considered to obtain an efficient rhizobial inoculant. And what we did was the identification of candidate genes involved in high competition abilities, performing a genome-wide association analysis. In the meantime, we also performed the isolation, characterization, and selection of Rhizobian strains with enhanced tolerance to salinity. So to perform the genome-wide association analysis, we first analyzed the competition phenotype, performing a nodulation test using different combination of co-inoculation with different Cynorhizobium meliloti strains, whose genome was fully sequenced. Why Cynorhizobium meliloti? Because it's one of the best biotechnological models in the study of the symbiosis with alfalfa, but in general for the study of the genetics of rhizobia. Each co-inoculation that we perform, we use two Cynorhizobium meliloti strains that were tagged with two different fluorescence proteins, the green one and the red one, as also you can see here in this beautiful picture, because we are very proud of this picture, actually. And after 28 days, we evaluated the nodule occupancy of the tested strain. And indeed, here in the graph are reported the red and the green bars, which are the percentage of single nodules, while the yellow bars indicate the percentage of mixed nodules, so nodules that were occupied by both strains. So in the genome-wide association analysis, we used this competition data together with the strain genomic sequences to identify the genetic biomarkers that control the competition and that can be used as markers in the further inoculant selection. And what we found is that multiple genes and functions are linked to this complex phenotype, at least in Cynorhizobium meliloti. And most of the genes that are involved in high competition abilities were mainly located on the symbiosis megaplasmid PCMA, which is a replicant that in Cynorhizobium meliloti carries all the genes related to the symbiosis. Some of the genes that we found codify, for example, for ABC type transport of amino acids and sugars, but also enzyme involved in the biosynthesis of cofactors, but also proteins related to other metabolism as amino acid degradation and carbohydrate metabolism. So to select the rhizobian strains with enhanced tolerance to salinity, we proceed with the second task. And thanks to the effort of our partner, of Professor Becchi and his collaborators, we isolated 39 rhizobia isolates from nodules of alfalfa grown in Algerian soils that were characterized by different salinity. And performing the 16S sequencing, we found that almost all strains were attributed to the Cynorhizobium meliloti group. Then we check if our Algerian strains may also have high competition ability. And to answer this question, we perform specific PCR using specific primers designed on genes that control the competition we found in the previous analysis. One genes indeed codify for this ABC type amino acid transpose system, while other two genes belong to an operon that is involved in the synthesis of the cofactor F420. Unfortunately, we didn't find this specific gene in our isolates, but we do not exclude the presence of other genes and functions among those that we identify that are related to the competition. So we decided to focus our activity mainly on the selection of isolate strains for the formulation of rhizobia inoculants, mainly use one criteria, which is the tolerance to salinity. And indeed, we perform an experiment. We trying to characterize our Algerian strains for their salt tolerance through an in vitro screening with the biotechnology. The biotechnology allows to record the metabolic activities of bacteria that grow on different sources and or different media, as in this case. And indeed, the Algerian isolates and 29 well-characterized collection strains were screened for their metabolic activities on increasing concentration of sodium chloride. And what we found is that, in general, Cynorhizobium meliloti differ in salt tolerance. Specifically, at 400 millimolar sodium chloride, we observed a significant reduction of metabolic activity in most of the strains, Also, at 600 millimolar sodium chloride, there were stronger differences among strains, and indeed the strains can be divided into 8 different groups according to their reduction of metabolic activity. But most important, we found 6 cynorrhizobiomeliloti strains that were highly tolerant to salt stress, showing the highest values of reduced metabolic activity at 600 millimolar. From now on, our activities were mainly divided in 2 sections. The first section was, has the aim to examine the symbiotic efficiency under salt stress. And indeed, we choose 38 cynorrhizobiomeliloti strains, both the collection and Algerian strains, that has different salt tolerance, and we evaluated their symbiotic efficiency under salt stress. And also, in the meantime, the 21 Algerian isolates were chosen and their genome were sequenced. So we started with trying to evaluate the different Medicago sativa cultivars and their ability to tolerate salt stress. Specifically, we evaluate the germination of 4 Medicago sativa cultivars under increasing concentration of sodium chloride. And we see that the cultivar marina and the Etrusca display high ability to tolerate salt stress, while the Maraviglia and Messe show an inability to tolerate high concentration of salt, at least at the germination stage. So we proceed trying to evaluate the effect of increasing saline stress on the growth of the cultivar marina and Etrusca. In this experiment, both cultivars showed moderate ability to tolerate the salt stress at 100 millimolar sodium chloride. So this concentration is indeed a threshold for the plant growth. In the cultivar Etrusca, we observed a significant effect of salt stress in decreasing the plant growth, especially for higher levels of sodium chloride, while the cultivar marina display a higher resistant and resilient to increasing salinity up to 400 millimolar sodium chloride. So from these 2 experiments, we observed that the cultivar marina and Etrusca show high tolerance towards the sodium chloride for both the germination and the growth test in the greenhouse. So to select, we evaluated the efficiency of the symbiosis between cynorrhizobiomeliloti and Medicago sativa cultivar under salinity. In this case, we use 38 cynorrhizobiomeliloti strains, both collection and the Algerian strains, in association with the cultivar marina and Etrusca. We also choose the concentration of 0 and 100 millimolar sodium chloride because, as I said, 100 millimolar is a threshold for the plant growth of Medicago sativa. And we see that an increase of plant growth parameters under salinity was promoted by several Rhizobia strains, and also that the strains able to significantly improve the dry weights of plant under salinity are different and specific for each cultivar. Indeed, here in bold, there are the strains that match in a positive way with the cultivar marina, and that Francesca is going to talk to you about later. So I let the word.
Thank you. Hi, everyone. I'm Camilla Fagozzi from the Department of Biology of the University of Florence, and I also collaborate in this project as well as in the Leguminous project. So for the second part of this work, of this task, we evaluated the, we went on characterizing these 21 cynorrhizobiomeliloti isolates from Algeria, choosing both salt-tolerant and not salt-tolerant isolates. And we performed genome sequencing with the Illumina technology by obtaining some data that confirmed that these strains all belong to cynorrhizobiomeliloti species. In particular, the average data obtained from the genome sequencing confirmed that the strains Arsenorezobium meliloti, and the phylogenetic tree that you can see here on the right shows the DDH distance. This analysis was performed with the TYGS service. And in this analysis were included the 21 isolates from Algeria, as well as 28 isolates from our lab collection. We also calculated the pangenome of the 49 strains, and here on the left you can see the pie chart indicating that, as expected, most of the genes that we found belong to the accessory genome. We have a large accessory genome with more than 21,000 genes. And all the strains resulted divided into two main clusters, partially similar in both the trees that you can see here. On the right, we have a core gene phylogeny obtained from a concatenamer of more than 2,800 genes. And on the left, a clustering of the dispensable genome based on the presence and absence of the accessory genes. One of our questions was about the competition abilities, and we asked if these 21 strains from Algeria have high competition abilities. So we analyzed data obtained from our genomes, searching the genes identified in our analysis and strongly associated with competition abilities. But as also Agnese mentioned before, we didn't find any of these genes, and this result was also confirmed by PCR. We found other functions linked to competition, but with a lower significant level. In our test, we included also the strain GR4, which is highly competitive, very interesting, especially for the in-field test. Our previous data highlighted the effect of symbiosis with the 21 Algerian strains and the effect of salinity on the growth of Medicago sativa cultivars, Marina and Etrusca. In particular, plant growth depends not only on plant genotype and strain genotype, but also on the interaction between strain and host plant genotypes, irrespective of soil treatment. However, the interaction between the two genomes resulted more relevant in unmasking differences in plant phenotypes, especially when plants are grown under salty conditions with respect with controls. This statistical analysis highlighted the importance of both the symbiotic partners and their interaction in the development of rhizobial inoculants. In particular, we know that symbiotic rhizobia could better impact over plant yield in salinity condition when associated with the salt-tolerant plant genotypes. And also that the development of novel cultivars resistant to harsh environmental conditions, as salinity, of course, should consider the use of symbiotic rhizobia during the various stages of screening. We have this part of work which is still in progress, and it's about a genome-wide association analysis aimed at identifying genes associated with salt resistance in Arsenorezobium meliloti. The approach that will be used is the same that we used before to identify competition-associated K-mers. We will match together data obtained from the in vitro screening, so with the biolog, and data obtained from the sequencing of the genomes of the 48 strain to identify sequences related to salt resistance. The tool that will be used to perform this analysis will be again Phenotype Seeker. In this way, our final goal for this task at least, it is the selection of symbiotic pairs, so rhizobium strains and host plant cultivar, to be used in field trial. For the moment, the cultivar Marina, which gave us very good results with, in condition of high salinity, will be employed in Algeria, Morocco, and Italy. And we identified the 5 Sanorhizobium meliloti strains chosen for their high symbiotic efficiency in presence of salt, evaluated again at 100 millimolars, that will be tested in field with the cultivar marina as associated plant. So thank you very much for your attention. Thank you for being here, especially for the people from abroad and Italy, of course. And thank to all the people that joined this group for this part of project. Thank you.
Thank you, Camilla. We were in time, so we have time for more than one question. Please, come here.
For me or Agnese?
Agnese. Agnese.
Both? Oh, okay.
Hello.
Okay, let's see.
I'm Elisa from Sant'Anna School, and I have worked on cultivated biodiversity, and also we observed mycorrhiza development with diversified lentils genomes. And I was wondering if you were speaking about the interaction of the Rhizobia strains and their effect, and I was wondering if you observed or you know of some effect of the interaction of the plant genotypes, so like intraspecific diversity, on the interaction of Rhizobia strains.
Yes, I mean, we know that, we know that different plant genotypes can develop different phenotype in terms of height, growth, biomass, number of nodules were inoculated with different strains, with, as I said, different effect when considered both combination of Rhizobia and plant. So the same Rhizobia with different plant has different results, as well as the same plant with different Rhizobia. We have a messy situation.
Yeah. But considering, like, have you ever considered, or do you know of studies about the effect of growing together different plant genotypes, and if there is an effect on the Rhizobia strain?
Growing together in the same pot?
Yes, like one next to another, if there are...
Okay.
Okay, any more questions? I have a question concerning the salt tolerance. In my experience, strains that are tolerant in vitro on plates sometimes do not correspond to the tolerance in the field. Mm-hmm. What's your experience in that? So how do you manage this possible problem?
Francesca is going to show you actually in field 4 strains that we selected that are in vitro highly tolerant. Actually in field doesn't work. But even in vivo with in the symbiosis with the plant have not so great abilities. I mean, at 100 millimolar sodium chloride, which is not a salt stress, but it is a salinity condition, and no, there is no
okay, thank you.
relation.
Okay, if there are no more questions, I think we can move to the next all-in project presentation. We have 4 speakers. Francesca De Corosi, Abdelkader Becchi, Khalid Azim, Maida Afidi, all these people are talking. Anyway, 20 minutes. Please, Francesca. Siamo online, com'è? some of the speakers will be online, so we need to set up the connection.
Good afternoon. I will show you the result of inoculant production and in-field trial in Italy. Mi scorre. Non mi scorre.
Perché questo è indietro, questo è avanti.
Okay. The aim of the in-field trial of the All-in project was to test Synorhizobium meliloti inoculants for boosting alfalfa production in the three countries, Algeria, Morocco, and Italy. Considering the concern about climate change and soil salinization, we choose the cultivar marina, since according to import experiment, it has an high tolerance to salinity. And about the strain, we choose 4 different Algerian strains isolated from moderately saline soil in Algeria, and that according to import experiment are able to increase the the marina growth under 100 millimolar sodium chloride. Another strain tested was the strain GR4. It is a strain collection that is able to efficiently compete for the establishment.
No tenemos sonido.
For the establishment of the symbiosis with alpha-alpha in lab condition. The experiment design was the same in all the 3 countries. It was conducted according to a randomized block design. We had an array of 7 by 4. We have 7 replicates and 7 treatment and 4 replicates. The treatment were marina seeds inoculated with the 5 different strains, not inoculated seeds as negative control, and not inoculated seed, but sown in pot amended with the chicken manure as positive control. The seed inoculation was performed in Italy. We use a carrier to transfer bacteria to the seeds to reach a density of 10 to 5 cells for seed. We choose the carrier among 3 different substrates, in particular perlite, compost, and biochar. We tested the survival of Synorhizobium meliloti in the 3 different substrates, and what we found was that perlite, but especially biochar, induced a reduction of the survival of Synorhizobium meliloti over a period of 19 days, while, on the contrary, compost did not affect the survival of Synorhizobium meliloti. For this reason, we choose compost as carrier for the production of the inoculant. The inoculant, the inoculation of the seeds was performed here in Italy, and then the seed was sent to our partner in Algeria and in Morocco for the in-field trials. In Italy, the experiment started on 20 of April, and the location was San Casciano, very close to Florence. We did not irrigate the field, and we also control the growth of the weeds. The end of the experiment was the 5th of October. In this slide, you can see the maximum and minimum temperature and the rainfall of the period. And as you can see, the temperature was generally high and the rainfall were low. At the end of the experiment, we collected the crop and valued the productivity, both by wet weight, measuring wet weight and dry weight expressed as kilogram per hectare. What you can see is that The strain that mostly increased the growth of alfalfa in respect to negative control was the GR4 strain, reported in green, that, as you know, it is a collection strain with that is an efficient competitor for the establishment of the symbiosis. About the Algerian strain, we can say that in our condition, they were not able to increase alfalfa growth, and this could be due likely by two main factors. First of all, that maybe the strain were not able to compete with the indigenous population of Rhizobia. We value the density of Rhizobia in this soil by the NPM test, and we found that it is about 10 to 5 cell 4 gram of soil. And furthermore, we have to consider that this soil is not proper a saline soil, so we can speculate that this strain should improve the alfalfa growth under saline stress, but not in our soil that is not a saline soil. At the end of the experiment, we also collected some soil sample to perform the 16x metagenomic sequencing to value the soil bacterial diversity. And what we found was that the inoculant did not influence the structure of bacterial community of the soil. So thank you for the attention, and now I give you the word to Professor Becky. Your presentation.
Thank you very much. I am Professor Becky from Algeria, and I am very happy to go to Florence for the third time. And my team collaborate with the team of Florence from the Professor Carlo and Professor Federico in two projects, All In and Leguminose. Here I am not very much time for your presentation, all the results, but I will present only the experimental trial result. You look here that our team is in Oran, University of Oran, and Laboratory of Biotechnology and Rhizobia, and we have this, our doctorand who work on this project, Amina. The experiment was begun in April. The sowing process of Medica gustativa marina seeds began. These seeds were inoculated with various strains of Rhizobia militae and carefully sown into their assigned plot. We observed the emergence of tiny seedlings on May 21. You look here, the experiment seeds before and after. We are also make the harvesting of the sample of soils. We observed the emergence of tiny seedlings. Aspect of, you look here, the aspect of experimental seed on the, after 4 months, 6 days before collecting the results. Aspect of Florence of the seeds before the harvest, measuring the plant height, aspect of the nodule still attached to the root as observed on the field. We are also collecting the stock of the plant biomass from experimental seeds that will later be used for the weighting of the dry weight aspect. You look here in this slide, the aspect of the host plant and the root nodule of strain MA, this compared to the positive control. We are also measuring the height Yes. You look here in this slide that there are a difference between the strain in the development of a height of the host plant. Principally, in the dried weight of host plant, you look here, the good strain is the MO15 and also the MO65. In the same time, also we look that the strain is very good in the development of the number of nodules. You look at also the weight of nodules. And you look here that GR4 is also a good strain. In conclusion, despite the exceptionally adverse weather condition experienced in this year, including cooling temperature reached up 40 degrees Celsius in July and August, we successfully maintained the Medicago sativa cultivar marina coupled with various rhizobia. However, it's worth noting that the height of the plant measured in terms of dry weight, nodule count, and nodule weight displayed variation depending on the specific strain used. Notably, the two strains, MO50 and GR4, exhibit the most promising result, delivering the highest yield among all the tested strains. Following the collaborative discussion with our agricultural partner, we recommended the adoption of strain MO15 and GR4 as the preferred inoculum for Medicago sativa production. These strains have demonstrated their resilience and potential to thrive even under challenging environmental conditions, promoting a more robust and productive cultivation of Medicago sativa. We also, in deliverable, all these results were exposed in many meetings. Here you look at with Algerian television and the regional radio. We also present this strain to our partner. You look here, the agricultural partner. This work would never have seen the light of day without the help of our doctorand and master student, as well as our agricultural partner, whom we thank for their precious help and their availability. Thank you for your attention. It's very, very difficult for me to speak easily in English. Thank you very much. I also add that I have many results, but we have not time to expose our, we have only 5 minutes.
Okay, thank you very much.
Thank you, Abdelkader.
And I think we go on with the other speaker of this part that are online, I think. It's Khalid Azim and Majida Afidi. Carlo, lo puoi collegare?
Can you hear me? Hello? Can you hear us? في المساج بقول can you hear us? Laissez au champ l'institut, appelle-moi. L'institut, laissez au champ des derniers. I feel trio, the same. Okay, ça fait maintenant, ok. Ah, donc كيف بس؟ Hello. Hein? Donc
Majida?
Yes, Carlo, how are you?
Okay, fine. Okay, you can start with your presentation. Please share your presentation.
Okay.
Click on the arrow.
La présentation, c'est pas la présentation. Okay. You hear us?
Yes, yes.
Okay. Well, hello, dear
Perfect.
colleagues.
Okay.
Happy to see you online in these scientific events, which success. We would have liked to be influenced, but outside constraints of time do not allow it. It's Nizar, PhD students who will present the test in the field present on molecular characterization and genetic diversity of rhizobium moderating alfalfa in Morocco, especially the field trial. Thank you very much. Can you hear me, Mr. Kaur?
Okay.
My name is Battas Nezar. I'm a PhD student. I have done most of the work for the All-In project in here in Morocco, including the communications and the translation of the video that you have previously shown. Project team members are Majida Hafidi, Dr. Khalid Azim, Dr. Habbadi Khawla, and myself. I'm PhD students. My thesis is molecular characterization and genetic diversity of rhizobium modulating alfalfa in Morocco. Today I'm going to present you the current accomplishments and future prospects of Project All-In. So the project aims to study the genomic diversity of rhizobia and its competition. Its objective is also researching the stimulatory impact of rhizobia on alfalfa in harsh conditions, the in vitro testing of different diets to optimize the use of alfalfa in combination of locally available byproducts, and finally the exchange of knowledge. So in Morocco, we have done a field trial in the region of La Nosar. It is in the western region of Fes, to the north of Morocco. It is known for a temperature that is averaging 25 degrees Celsius with quite low precipitation. We have We have conducted the experimental design in accordance with the Carlo-Vitti protocol with very few changes. Number one, we did not inoculate the strains more 1, more 2 because they were not ready at the time. And the distance between blocks was reduced from 1.5 to 1.2 just to minimize the size of the scale of the project. The plot was also regularly monitored. We did use irrigation. This is the date of sowing. This is the 23rd June 2022. We have sown late. We also added irrigation 2 days later. This was the early signs of growth in all of the blocks so far, which was on the 21st of July. We have noticed that there was a lot of weed at the time, so we have devised to manually remove all the weed on the 30th of August. And finally, the first cut was conducted on the 22nd September, and the second cut was on the 26th December. These are the analysis of the field soil. The rhizobial count was estimated to be 2.09 10 to the power of 5 for both depths. The depths from 0 to 20 centimeters and 20 to 40. There is the soil is called hamri. It contains mostly clay and large rocks and very little silt. For the results, these are the plant's mean heights in centimeters during the first and the second cut. The lowest ones for the first cut was the negative control and strain MO56. And for the highest, it was the positive control, which we have added nitrogen to, whereas the other ones, we did not add any nitrogen at all. Only the bacteria that were already pre-inoculated in Italy. For the second cut, the lowest was recorded for MA10. It was even lower than that of the negative control. And the highest were those for the MO15 and MO35 strains. For the plant mean wet weight, the lowest was recorded for negative control and GR4, which is the Italian strain, but the highest was the one from MA10. Conversely, it was also the one that scored the lowest on the second cut, as well as the negative control. The highest at the time was GR4, so we had a complete switch in terms of wet weight. This is the wet weight compared to the dry weight in percentage. The lowest was recorded for MA10, and the highest was for the positive control. The lowest on the second cut was the strain GR4 and the positive control, but the highest was MA10, which was the opposite of what we found earlier again. For the mean nodule weights, we have found that the lowest weight was for the negative control, but oddly enough, we found the highest to be for the positive control, meaning that not only did it nodulate, but also nodulated more. The mean nodule count was lowest for MA10 and was the highest for the positive control. However, it was the positive control that had the lowest weight per nodule compared to the rest. So it had the lowest weight of the nodules, but the highest count of the nodules. The highest recorded was MA10. Our results thus far are still inconclusive because we still have other tests to conduct, especially the one for nitrogen to have proper final conclusions. But thus far, the strain MO35 and MO56 have shown to have the best results overall compared to the rest. The difference between the positive control and the MA10 might be linked to the fact that the soil already contains a certain amount of rhizobia, so maybe those rhizobias were actually better than the ones that we have tested previously. in addition to the tests that we have conducted in the field, we have also conducted a field trial in the National Institute of Agronomic Research of Meknes. We have sown 63 pots on the 17th February of 2023. The germination occurred on the 24th. The first true leaf, which is the leaf that contains 3 folioles, was recorded on the 10th of March. We have thinned the number that we have used. We planted five seedlings each, and then we have thinned them to only just 1 on the 3rd. Finally, we have conducted the cut on the 3rd of July, 2023. For the protocol, we have actually used the Itaka variety, which is the locally sold variety in Morocco, which to my understanding, it originates from Italy. These seeds were inoculated with 10 to the power of 9 colony-forming units of bacteria that we have isolated from the regions of Agadir and the regions of Meknes, which are arid and semi-arid regions. We have thus far collected approximately 90 strains. We have only tested about 6 of them in this experiment. We've grown the bacteria on yeast mannitol medium at 27 degrees Celsius for 2 days with constant shaking. Then we have centrifuged everything. Then we resuspended the bacterial culture on...
Excuse me, you have 2 minutes to finish. Go to the conclusion, please.
Okay. No problem. These are the results of These are the results of the greenhouse trials. We have had 3 different irrigation types. We had 100% of the pot's capability, 66% and 33% of the water. We had positive control and the negative control as well. What we have noticed is that the ones that had the highest amount of water have shown to have the biggest height. However, in terms of the number of branches, we have seen that the 66% was the one that had the better production, as well as for the leaf quantity. So far, the strain that has shown to have the best results was the strain B, which came from the region of Agadir, the region of Ait Milk. That's the name of the region. And we have also found that the 100% water that we have provided to the plants was actually more of a stress to the plant rather than providing it proper water. So 66% was actually the right amount. For future works, we are trying to do in vitro cultivation of alfalfa. We're going to either use the Morashi-Geskog medium, but without nitrogen, or the Johnson medium, depending on what we have available. Our attempt here is to try to remove entirely the field trials and just use either greenhouse testing or in vitro testing in order to identify whether a strain has the capability or not. So we're trying to do a comparative result. And among the future works is also the genetic diversity, which is part of my PhD. So genetic diversity can be studied using housekeeping genes such as REC-A, RPO, NOD-A, NOD-B, NIF-H, and so on and so forth. Finally, to conclude, a significant progress was done so far towards the sustainability of alfalfa in the Mediterranean region, and all-in has been a major element in this achievement. However, the journey towards sustainability of alfalfa does not end here. And thank you for watching.
Thank you for presentation, all the speakers, of course. Unfortunately, we're a bit late, so is some burning question? No question. So thank you again to the speakers, and we move to the next presentation. Is again all-in project. Marcello Mele, Arianna Buccioni. Work package 2, circular economics in ruminant diet in vitro and in vivo trial.
No? Okay. Good afternoon. Now me and Giulia go to present the result in the activity of the work package 2 about the in vitro and in vivo trial where we test alternative ingredients in dairy sheep feeding strategy. According to the objective of FAO 2050 diary, we choose to test several alternative ingredients, several from byproducts of the agro-industrial production chain and other from the autochthonous culture of the marginal area that are cultivated in the Mediterranean area. As well as these are rich in functional molecules such as polyphenols and fatty acid that can modulate rumen metabolism and contribute to improve the quality of the end product of the ruminant production chain. So the in vitro rumen liquor fermentation was conducted with an in-batch fermentation, lasted 24 hours, and singularly the alternative ingredients were tested. At the end, the rumen fermentation, 2 aliquots were collected, one for the fatty acid profile characterization and the other one for the rumen microbiota determination. And the volatile fatty acid profile was used for the methane potential production prediction. Here I reported the approach that we usually use for the DNA extraction and then for the data acquisition. This is the control diet that we use and the result about the fatty acid profile. As we can see, olive leaves, olive oil pomace, tomato peel, rice bran, and olive oil pomace ensiled modulate in different way several unsaturated fatty acid concentration. With citrus pulp, tistle panel, and grape mark modulated fatty acid isoform in different way too. About the methane potential production, we can see that olive oil pomace, cardomeal, and grape mark mitigate the methane potential production that we estimated. And about the rumen microbiota, we can see that olive oil pomace, olive leaves, and ensiled olive oil pomace didn't affect the alpha and beta diversity of the rumen microbiota. So they was the control and the treated fermenters were similar, and the difference into the diet did not observed. And the genera affected were different for the different alternative ingredients used. So from the in vitro trial, we can conclude that the products tested affected in different way the several fatty acid, and several of them affected the methane potential production in several in different way. Interesting data about tomato peels, rice bran, and olive oil pomace were observed on rumen metabolism. And even if no full data are still available about the rumen microbial communities, it was formulated a treated diet with olive oil, pomace, rice bran, and tomato peel as alternative ingredients to test in vivo. Thank you for your attention.
So good afternoon, everyone. Thank you for being here. So during this second part of the presentation, I'd like to share with you the outcomes of the second part of the work package 2, and which was carried out by the group of the University of Pisa, and which aimed to valorize the inclusion of byproduct in vivo ruminant diet together with the alfalfa. So very shortly, a background related to the positive effect related to the inclusion of a byproduct in ruminant diet. So firstly, their use satisfied the principle of a circular economy supported by FAO, but also their use minimize their impact otherwise due to their disposal. They represent alternative sources of nutrients, and in this sense, they can help in reducing the feeding cost. And also, they deliver some secondary metabolites. We can have an effect at the animal level, such as can help, for instance, in improving nitrogen retention at rumen level, but also, as shown earlier, in reducing methane and so on. But also, they can have some positive effect at animal products level. So, for instance, on milk and meat quality by improving their for instance, for the fatty acids contained. So what was done previously was to conduct a surveys in all the countries involved in the Lean project, but also to consult the public inventories in order to understand which byproducts are more present in these countries and for which they can be interesting and an alternative use. So after such consideration and also after the in vitro fermentation, as shown earlier, we removed some of these byproducts and then we add rice and wet bran as a source of starch because most of them are quite lack in starch. So, sorry, what was the idea? The idea was to consider all these byproducts all together and try to valorize the peculiarity of each byproduct, to mix them and to produce a meal in order to be fed to ruminant. So the in vivo trial was conducted at the Department of Agricultural Science of the University of Perugia in Italy with the dairy sheep. The breed was Comisana. You can see a picture here. We had, the trial was conducted this year. We had two groups, the control group and the byproduct group. The experiment lasted in total 6 weeks. So basically, the experimental conditions were the same for the two groups, and the groups were distinguished only according to the diet fed. So they were fed with two different types of meals. One mixture meals made by byproducts. So you can see here in the pie chart on the left, we had olive pomace, rice bran, wet bran, distillers of wet, tomato peels, but also alfalfa meal as a source of protein. And so all of the byproducts are particularly not in competition with the human nutrition. On the other hand, we had the control mixture meals, which contained the soybean meal, corn meal, barley meal, which are more meals more in competition with the human that are traditional use in ruminant production. These two different types of meals were fed with the alfalfa A, and also animals were grazed on open pasture. So as anticipated, the trial lasted six weeks. The milk was collected each week to be analyzed for milk composition, and also milk production was recorded daily. Also, rumen fluid samples from the animals and also other measurements related to methane, enteric methane production were analyzed at the beginning and at the end of the experimental period. So moving to the results, very interestingly was the fact that milk production did not differ throughout all the experiment between the two groups. So the higher inclusion, so the particularly high inclusion of byproducts, and which was characterized with a very high level of fiber in the diet in contrast with the low content of starch, did not cause a decrease in milk production. Also, milk quality was not particularly affected. Fat concentration remained similar. Only the protein were lower in the case of byproduct milk. But on the other hand, the milk clotting aptitude seemed similar between the two groups. Urea, as you can see in the graph on the right, decreased accordingly to also to decrease of protein and the byproduct group in orange. And this can suggest a lower degradability of, as expected, a lower degradability of the protein contained in byproduct in comparison to the other traditional meal. So moreover, also regarding the quality of milk, also the milk fatty acid were analyzed in all the samples of milk taken each week. Mainly the milk from the control group show higher concentration of the novosynthesis fatty acids, and also higher content of saturated fatty acids, and higher content of odd and branched fatty acids. On the other hand, the milk of the byproduct group show higher concentration of the intermediates of biohydrogenation of stearic acids, and among them also some beneficial fatty acids for human health. Results on milk fatty acids of the control group were also confirmed was supported by the volatile fatty acid profile analyzed in the rumen liquor taken at the end of the experimental period, in which was found a higher concentration, higher molar proportion of butyrate, which is the precursor of the production of fatty acids up to 16 atoms of carbon, so the palmitic acids, meaning suggesting a higher activity of the fatty acid synthetase for this type of milk. In the case of byproduct milk, the higher accumulation of intermediates of biohydrogenation seem more related to the higher concentration of linoleic acid in the byproducts, such as olive pomace or tomato peels, which led to higher accumulation of such compound. And among them, as anticipated, there are some with, for instance, CLA, with interesting properties for human health. So another question was related to methane, because we tested particularly higher fiber content diet, which is indeed less in competition with human, but still contain a higher content of fiber. And this, we know, can cause higher production of enteric methane, which is something we did not want to increase. And very interestingly, by comparing the two groups, you can see the histograms on the left side, we did not find the differences between groups. So meaning there was not higher production of methane in the groups fed with higher fiber content in the diet coming from the byproduct. So in conclusion, by mixing all the byproduct listed earlier in a meal for ruminant, Those diets are characterized with a very low content of starch and a high content of fiber, and also the protein is expected to be less degradable in comparison to more traditional feed. But this type of meals are less in competition with the human nutrition. And thanks to the rumen, it's possible to feed them the ruminants and to valorize, because they are able to convert the fiber material and to produce energy for their normal metabolism, as well as methane, as we have seen earlier, was not difference between the two groups, and to produce milk, which did not show a different production between the two groups. So it was possible to produce a similar, have a similar performances between the two groups. And another aspect, the last aspect that I wanted to highlight was the protein contained in milk are of high biological value. So we started with a very low biological value in the byproduct, which cannot be used for human health, and then we got an improved quality product, together with a higher concentration of beneficial fatty acids. So we want to conduct further study on ruminal microbiota in order to understand the pattern occurred in the rumen and to understand better such results. And so for now, I'd like to thank all the part and all the group. Here in the picture is the group from Pisa, but I'd like also to thank the group from Florence and the group from Perugia for their help with this trial. Thank you.
Thank you very much. You have not been introduced because your name is not...
Clearly, we are not Professor Mele and Professor Buccioni. So
that's realized.
Yes, I am Giulia from the university, from the Department of Agricultural Science of Pisa.
We are in time now. I think we can have some questions, if any. Speaker.
For Federica, maybe.
You found a reduction of methane emission using some byproduct. Do you have an idea about the effect of this byproduction on methane reduction?
We estimate the methane production potential with the stoichiometric formula, and we on the bibliography is frequent that these alternative ingredients mitigate the methane production. So probably we could estimate
the molecule involved are the polyphenols or other...
with the probably are polyphenols, the polyphenols, yeah. That are in the alternative ingredients.
Okay, thank you.
Maybe I have probably a naive question. All this material that you use as alternative for feeding is cheaper than the regular feed?
So for some of the byproducts shown in the pie chart earlier, there is a price on the mark. For other, there's not. So it depends also on the strategy used to improve the shelf life of the byproducts. So, for instance, if they needed to be dehydrated or needed to be acidified and so on, and to the transport connected to the use. But basically, the concept is for most of them, the industry which produce byproduct have some cost related for their disposal. So they want to find alternative way to use them.
Okay, thank you. Any more questions? No, I think we can go on and with the last presentation for All In project, Alessio Mengoni. work package result outreach.
Okay, good afternoon, everybody. I will try to be short because we are in late and at 7 they close, so we need to move on. So my task now is to show you which have been the dissemination activities of the All-In project. Sorry for the work there. Of course, you know what the All-In is, so we can just skip. There are 3 work packages, and one was devoted to cooperation among countries, meaning that within the aim of this funding scheme, there is not only science, which is important, of course, but there is also, and especially, cooperation among countries and dissemination of the importance of the research, which has been funded by the society, because we are using public funding. So, we had, within All-In project, several associations and stakeholders which contributed in helping the dissemination of the project. You can see here some of the stakeholders in the association in Algeria and in Morocco, for instance. And within this work package 3, we can define some aims, 4 listed here. Of course, dissemination, but also communication, and especially educational activities. Of course, then, at the end, we would like, as we discussed this morning for people which were here, in Italian, sorry, to go to the market. Of course, we cannot go to the market directly, but we need to have in mind that there is a market in front of us. The market meaning using, in this case, bioinoculants and the technology development in the project for decreasing input in agriculture. So dissemination and communication activities, of course, involve a website which was created, a social media, and there was recently a tweet by somebody of you here, and dissemination to professionals, because we need to teach how to do and the importance of bioinoculants. And this can be done in synergy with the professionals. It is not just going from lab to the professionals, but a tight interaction. And this was done within this project. And then, of course, we are doing science. Then dissemination as the classical ways, meaning publications of conferences, to scholars, to professionals as us, as scientists mainly. But then, of course, it is important that young scientists, young researchers are involved. Young researchers which can be researchers in the academy, but especially, and more importantly, in the private sectors. So people, in the case of All-In project, which can develop novel bioinoculants, develop formulation of bioinoculants, and what is included in all this panel of exploitation of results. And then outreach. Outreach is meaning that we need to go beyond the professionals, beyond people which are really interested into, because we need to let know the society the importance of this kind of work, because we need to keep in mind that we are using public money, and public money are taxes from people, from all of us, and people from the street behind. So we need to show to them which is the importance of what we are going, and contribute in creating novel generations, not of scientists, but of citizens which are aware of the importance of decreasing the input in agriculture, promoting sustainable solutions, and so on. So a lot of activities have been done in this kind of area. of course, we had a mailing list, and we participated to several meetings, of course, the meeting of the project, but also other meetings as workshops to professionals from the Accademia dei Georgofili, one of the most important academy in Italy for agriculture, which is here in Florence. We are proud of that. And then formation of young researchers, so a summer school which was organized in Florence. And especially, I like a lot outreach activities. So several outreach activities you can see in this list, from activities in the primary school, so with the kids. Scienza Estate is another activity which is routinely carried on here in Florence, and then workshop, and then the night of the researchers, and so on. Probably I have not listed all of the activities we have done. We just to give you an idea of the kind of activities which we are meaning as outreach activities in this project. The website, you can go on in the website and browse the website. This is the workshop we have contributed to. It was in Italian, because this workshop was involved with the Italian stakeholders and farmers especially. So it was attended by more than 400 people. And the focus was nitrogen fixers, both free-living nitrogen fixers and symbionts. So of course, we were quite engaged as all-in partners within the preparation and dissemination of results to farmers within this webinar. And then technology transfer. Carlo mentioned it this morning that the seeds we were using in All-In project were contributed by a company in Italy, Continental Semences SPA. Continental Semences is the business of Continental Semences is a seed company, so they are producing seed, and they are producing coated seeds. So seeds of alfalfa and other pasture legumes, which have been coated with rhizobia and also mycorrhizae. And then they were interested in what we were doing, so they contributed with alfalfa varieties. Marina variety was contributed by them. And within this kind of activity, one of the PhD students we have in the lab, she is doing an internship with Continental Semences, because technology transfer is meaning also sharing young scholars to transfer the technology in reality. Some pictures, the summer school, the bright night, which was done in Pisa a few weeks ago by Marcello Mele's group, and then the Twitter account. You can follow it and see what we have done and we are going. And then, as also mentioned by Professor Becchi, a lot of other outreach activities and dissemination which have been done by partners. These are a couple of pictures which were shown by Professor Becchi also on interview, on local broadcast, which are important activities, because we need to let people be aware of what we are doing in the lab. We are not closed in some ivory towers, but we are doing something for the society. Other tweets, we were doing farming activity. These are the feed trials we did within our activity. And then I think this was, at least to me, my feeling, one of the most relevant results of the outreach activity, was mentioned by Carlo this morning, was a motion graphic video, cartoon. So in this motion graphic video, which is for kids, of course, you can watch on YouTube, you can browse it, and there is an Italian version, but also with the undertitles in French, English, and Arabic, I think, is going on. Why a motion graphic video? Because, you know, at least my son and daughter, now they are not anymore kids, but they like to play with a mobile phone. So without mobile phone, they are lost. And if you, instead of just writing and doing in-person activity, you have also some video to show them, maybe they can reach the point. And the point is to let know young kids that a plant is not living alone, a plant is not an island, because plants are rich of microbes, and those microbes are contributing to the benefit of the plant, and in our case, nitrogen fixation. And then, of course, papers, because we are scholars. So we publish it. We are quite happy to have a lot of publication within this project. Some of the results of this population was presented before during the presentation of work package 1 and 2. So we span from journals related to very basic and microbiology, like M-System, to opinion papers, which we published in Trends in Microbiology, related to a general view of the microbes and plants, because this is the, say, the central point of the project. And then several conferences, as Carlo and some of you mentioned before today. We participated to the European Nitrogen Fixation Conference with several contributions. This is the conference which is European, but indeed international, which is collecting the most up-to-date and important scientists in the nitrogen fixation area. Of course, we are not within the most important nitrogen scientists, but anyway, we were happy to participate in this conference. And this 15th edition was held in Italy, so it also easy to get there. And then other conference which we attended. So just to conclude, because as I told you, we are in late, so we need to run. I show you the website and the Twitter account, and so project is ended, but results are still ongoing. So be online to see what is going next. And thank you for your attention.
Project, and the leader of the project, Federico Martinelli, will show result on working package 1, phenotypic and molecular characterization of lentil and chickpea biodiversity, Mediterranean basin. Bye, Federico.
Okay, so I'm gonna briefly show you the results of our first work package that deals with the phenomic, genomic, and agronomic characterization of our germplasm of lentil and chickpea in Mediterranean basin. So, as I said this morning, this work package is composed by basically activity 2 and activity 3. In the activity 2, as you see, Scuola Superiore Sant'Anna is involved in the genome-wide association analysis in chickpea. In chickpea, we are in the middle of the work of identifying SNPs that are related with some important agronomic traits. So in this work, we are in program to do the sequencing of 90 genotypes, stable genotypes, and we have already the agronomic data in the field that was done in collaboration with Genetic Services, a company in Italy. And in these cases, so we have characterized in the field the 90 genotype that we are sequencing, and we have the data of the field. And as you see, we have done the analysis of some key traits of chickpea, and we have identified the most productive and the less productive genotype. So we will match this phenotypic data with the genomic data with the aim to identify SNPs related with important traits in chickpea. In lentil, the work is in advance. So this work basically has been already done, and the Scuola Superiore Sant'Anna have done the genomic and phenotypic characterization of more than 110 land races, and the Scuola Superiore Sant'Anna has already performed the data on the field. These data were able to identify the best performing land races in 2 years, in 2020 and in 2021. And an important finding was the fact that it was shown that landraces behave better than commercial cultivar in these 2 seasons that were characterized by harsh weather. So in the lentil, the genomic analysis has already analyzed, and some SNPs has been associated with some important traits like flower precocity, susceptibility to biotic stresses, and plant high. And as I said, the main results of this part were that there were some land races interesting that behave in a good way, and some SNPs were identified linked to these traits. In the activity 2, also, I am working on this aspect of drought transgenerational memory in collaboration with CNR, Francesco Loreto and Susanna Pollastri. And in this part of the activity 2, we are trying to get inside in the transgenerational stress memory in chickpea, transgenerational memory to drought. And here you can see our pipeline. Basically, first we wanted to screen genotypes in order to identify the 2 opposite genotypes, the tolerant and the sensitive genotype. And then we made the transcriptomic analysis in response to drought. And in the 3rd step, we are doing an experiment on the filial generation in order to investigate on the mechanism related with transgenerational memory. So we have done this trial, this simple trial, where we are basically comparing 2 conditions, the control, where we provide 70% of evapotranspiration through irrigation, and the drought characterized by 30% of irrigation of the evapotranspiration. And basically, so we are doing this in a controlled environment, and we are analyzing the response of the plant at different levels: photosynthesis, but biochemical stress markers, and transcriptomic analysis. As I said, in the 1st step, we tried to screen the genotype, identifying the tolerant and the more susceptible genotype. And as you see, we have done a trial on 9 genotypes, and we identified 1 genotype that reduced the chlorophyll content. This was indeed more sensitive to drought. And then we measured the osmolytes, soluble sugars, and proline, and we identify 1 interesting genotype that increased sugars and proline in response to drought, and also this genotype did not reduce the chlorophyll content. And so we choose these 2 genotypes to do the RNA-seq analysis in order to identify the up and down regulated genes in response to drought. What are the results? As you see here, we have done a gene set enrichment analysis comparing these 2 different genotypes. And some good results came out from this analysis, like the sensitive genotype, the degli, this degli genotype, increase genes involved in photophosphorylation, brassinosteroids, also cell wall modification, and also this BHLH transcriptome factor. Also, it was interesting that this sensitive genotype reduced genes expression involved in transport, solute transport. That's probably the main cause of the sensitivity to drought. And the other one, the most tolerant, as you see, did not regulate too many pathways. And so this was expected because this genotype was feeling better. But an interesting thing was the fact that upregulated the chromatin organization genes. So we believe some epigenetic effect are linked with the tolerance to drought. So we tried to summarize the results in this slide that we made. As you see, we tried to see which differences are between sensitive and tolerant. And as you see, the sensitive induce upregulation of many genes involved in hormone, like gibberellin, ABA, and brasinosteroids. And there is also upregulation of genes related, as I said, to cell wall modification. While in the tolerant, as you see, there are much less up and down regulated genes. Some interesting genes, as I said, on chromatin modification that were upregulated. So that's why we decided to focus on the next experiment that, as I said, deals with transgenerational stress memory. So we basically established a simple trial where we wanted to identify the differences in the F1 generation coming from parents that were not stressed. And we wanted to compare this one with F1 coming from parents that were stressed, okay? In the control and stress condition, again. So with the aim to see if there is a transgenerational priming to drought in chickpea, and which genes, which epigenetic modification are involved in this probably transgenerational memory. So we are in the middle of this experiment. We have done the collection of the samples. We are almost ready to analyze the transcriptome and also the methylome. And we are planning to do also, of course, the same analysis that we have done with the parents. So photosynthetic parameters, chlorophyll content, biochemical stress markers, and also we would like to do metabolome analysis. So we want to integrate the different omics approach in order to have a comprehensive analysis of all the key players that could be involved in this phenomenon. Because we saw the phenotypic data, I'm not showing, but we observe differences in the phenotypic analysis between F1 from controlled parents and F1 from stressed parents. They behave in a different way, and so this is interesting, and we want to, of course, identify what is the mechanism underlying these changes. In addition to this, also, we have established an hydroponic system to study chickpea and lentil, to try to, again, also investigate drought stress tolerance. And so we have established an hydroponic system using PEG to simulate osmotic stress. And here we have done transcriptomic analysis in response to drought in both chickpea and lentil. And we have done a real-time analysis for some genes, as you see. And some interesting genes were upregulated in response to osmotic stress. These genes were mainly well-known transcription factors. Some others, they were downregulated, and so they can be probably negative regulator of drought response. And the same we are doing for another important trait, branching. We identified two genotypes that were behaving in a different way for both chickpea and lentil. One genotype was not branched, and the other one was highly branched. And also in this case, we have done a transcriptomic analysis of apical shoots and axillary shoots with the aim to identify key genes involved in branching. And we have done also, as you see, real-time analysis in order to identify some possible genes that could be interesting, maybe for the future, for doing molecular breeding. And interestingly, we observe some transcription factor related with the strigolactones that are well known to be involved in branching. The activity 2 deals, as I said, also with phenomic analysis. And this work is done by Francesco Loreto and Francesco Cellini. Francesco Cellini is the head of the facility in Metapontum, where they have great infrastructure where they do high throughput phenotyping, and they can analyze leaf area, plant height, growth rate, and other parameters in a very high throughput way. And here we are planning in this month to do an experiment. The aim is to characterize this F1 generation. I will provide the seeds to him, and hopefully he can make a high throughput phenotypic analysis of this F1, so we can add another level of analysis in addition to the other omics analysis. And in conclusion, the other activity that is dealing with work package 1 is the activity 3, that it's the agronomic screening for drought tolerance. So in this work, that basically we have done before the activity 1 and 2, what we have done was trying to screen as many genotypes as possible in response to drought. So basically, from the germplasm that we have, we selected almost 100 genotypes of chickpea and lentil, and they were screened by different partners. 5 countries were involved in this activity. UNIFI, University of Oran, IFAPA, CBBC, and University of Sirnak. Basically, we have done a screening in controlled condition, but also University of Sirnak have done this in field condition. So basically, we wanted to identify the best genotypes for each country. That was the rationale of this activity. And so we analyze different traits, plant height, the biomass, obviously the yield. And in these cases, we tried to adopt the same protocol. Obviously, it was not easy because, as you can imagine, it's very difficult to standardize all the conditions between different partners of different countries, but we try to standardize as much as possible the protocol, and at the end we identified 3 best genotypes for each country, for chickpea and lentil, the one that they are supposed to be more tolerant to drought. And even if we had so many problems for, as you can imagine, to ship the seeds and analyze them between different countries, we tried to analyze these interesting genotypes with physical parameter, chemical parameters, and so we have done, as you see, all these qualitative analysis on these genotypes in order to characterize them also at qualitative level. And as I already showed, we are trying with a genomic approach to identify SNPs related with important traits using these genotypes. This work basically is done, the physical parameter and the chemical parameter by CNR. In this activity, as I said, we have done a part in controlled condition, but a part in the field. And in the field, as I showed this morning, the work was done by Daria in Turkey. and I already show you this morning that the work that they are doing, they are screening basically local genotype. Unfortunately, it was not possible to screen in the same environment these best genotypes, because the idea was to the best genotype that we identified to screen in the same condition, all, but it was impossible to do it, unfortunately, for reasons like, as you can imagine, shipping seeds between countries outside Europe and inside Europe is really, really, really terrible. But anyway, we wanted to do this for the local genotypes in Turkey, and I already have shown the results this morning. Thanks.
Thank you, Federico. We have time for questions on the presentation. C'è no question? Andiamo avanti, allora bisogna collegarsi con Nureddin. Next speaker is online, is Nureddin Yala.
Hi. 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 Hi.
Okay, we can hear you.
Hi, please.
Okay, Nureddin, if you are ready, can you hear me?
Yes, I hear you and I am ready. Thank you.
Excuse me, we cannot follow. We cannot follow the talk. We cannot follow. We cannot see the screen. We cannot see the slides. We cannot follow the meeting.
Guardare su YouTube, hanno il link su YouTube. Dovevano guardare su YouTube.
Well, you should have connected to YouTube. Anyway, now I think we have to move on with your presentation.
Sorry, me?
No, ma va bene tutto, però poraccio.
Come si fa? Cioè, dobbiamo andare avanti o no?
Sì,
Come si fa?
ora chi dovrebbe presentare dovrebbe condividere la sua presentazione.
Chi è che deve presentare?
Noredin, io non so chi sia Noredin.
Ma chi sta adesso?
Chi è Noredin? Ok, bisogna dirgli di condividere la sua presentazione. I know, the problem is that to see the slides, they should link to the YouTube link. They should follow via YouTube link, but I think that this kind of information was already provided. Yes, we can send it.
Can we start with the Dulce?
Calendario, no, su giù. È YouTube? Ah, il link YouTube? È sulla... Il link YouTube è sulle mail, sul gruppo di WhatsApp.
Aspetta, facciamoglieli proprio.
Qui c'è un'assistenza.
Ma
www.unifi.it/webtv.
Sorry for this inconvenience.
unifi.it/webtv.
ok, ora bisogna dire a quella persona che deve presentare se per favore condivide la
Chi
sua
deve presentare? Allora,
Ok,
Noureddin, are you listening?
Yes.
So you have to share your presentation. You have to share on Gmeet.
I tried. Federico, do you hear me?
You have to do it. You have to do it. You have to share your presentation on your computer. We are allowing you to do it.
Google Meet or by YouTube?
YouTube is just to see the presentation, but you need to present sharing your presentation. Clicking on the arrow, on Gmeet.
Meet, but
we are. Did you see this button here with the arrows? You have to click here and share your presentation like this. Okay? So now you see what we see on our computer. Okay? Okay. So you have to click into this button. Did you see it?
Please, shall I share my computer about Google Meet or by YouTube channel? Do you hear me?
Please send me your presentation at the address carlo.viti@unifip.it.
Okay, I try to do it.
Sì. Ma il dolce l'hai riattivato, dolce?
We can switch to the Dulce Rodriguez Navarro talk and then go back to Noureddin. Dulce is listening. Neanche Dulce sta sentendo.
No, è disattivata. Bisogna cercare il modo di attivarla. Forse bisogna chiedere all'assistente.
Ma se è fatta, il problema è qui. Please, Dulce, could you try to switch on your microphone, your phone? Come li si attivano?
Cristina, no?
Quale?
Tutti, attivali
su Cristina.
Deve essere un coorganizzatore che presenta.
Ma erano tutti attivi. Ok, perfetto.
Senti, io vado avanti con lei, poi dopo...
Ok, va bene. Aggiungi come coorganizzatore.
Okay, when we're waiting for the connection to establish, we can go on with an addition to the Noureddin talk that is by Annalena Bollerde from the Leibniz University, and she will have a short presentation of what she's doing in this. Gli possiamo dare lo schermo?
Sì, 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 ok. Però adesso li posso. Could you try to switch on the phone, please, Dulce?
Now it's on, and now...
Perfect. Okay, perfect. Okay. Can you send you? I sent you the slide. Ok, comunque io dovrei avere ora le mail. Qual è la? Ok. This? Ok, perfect.
Okay. Now that they're trying to establish a connection, we are starting with Annalena.
Yes,
okay,
so
please.
I'm going to present the results or preliminary results from activity 4, which is also part of the work package 2, and Noureddin is going to present the other results of the work package. And I'm going to bring in a quite new perspective from the rest of the group, because we were looking at the legume systems from a nature conservational point of view, so we are looking at the wild flora that is actually in the systems. So I just want to move the slide. That one. Oh, thank you. So the overarching question of our research was what factors actually do increase the associated biodiversity in lentil and chickpea fields in the Mediterranean. And by associated biodiversity, I mean the wild flora that is actually occurring there. Methods-wise, we were first conducting a literature review to see what's the current state of knowledge and also to retrieve indicators to look at. And afterwards, there was the big bunch of work we were doing, which was the field work. And to do so, we did a full vegetation survey of arable wild flora, where we were actually surveying the wild species that were occurring there and also the cover estimates. We did this by means of a repeated randomized sampling in 1.2 plots following the drilled lines of the experimental plots. And we conducted that in 2 seasons, in 2022 and this year, in the season May to July. And we were visiting 2 kinds of trials or areas, I would say. First were the experimental trials of our partners in Spain, Italy, Tunisia, and Croatia. And then also to further check and validate the species richness, we were looking at organic farms in Italy. After that, we did a post hoc survey to deduce explanatory variables, so to see how they were managed exactly, what was the weather and soil conditions. And then followed the analysis. We did a spatial analysis to look at the landscape context of the fields by means of the diversity of habitats in the surrounding. And then we were doing statistical analysis, looking at the Shannon index. We did correlations, and we used methods from meta-analysis to estimate the effect size, and still we are looking at analyzing the farm results, which I cannot present today because we are still into analysis, but this will be a redundancy analysis. So first, to give you an impression of how our sampling looked like, you can see that we had a bit more sample plots on the experimental sites than on the farms. And I'd like to, is the pointer working? No. Sorry. So here you can also see that it's important to also look at farmers' fields, because we can see that they have a significantly higher share or higher amount of wild flora species than the experimental sites. And also comparing the experimental sites, we can see that we have significantly differences amongst the countries. And now looking at the effects of different management practices, when you're looking at this diamond star, you can actually see that it is in the middle, so 0 means neither decrease nor increase. We don't have an overall significant effect. but it is important to look at the kind of management that actually has been applied, and then we can see that if we are testing for the subgroup difference, we can see a significant difference. Further going into this, we see, no surprise, no weeding is actually significantly superior to weeding practices, so you can see that the overall effect actually increases wildflower species richness. And as for the rhizobia inoculation, we don't see any significant effect, neither decrease nor increase, so it does not affect the wildflower species richness. And then for the cropping type, we can see here we don't have a significant effect, but we do see a tendency that monocropping is superior to intercropping. And then we were also looking at correlations, but I want to highlight that this is the data set from last year, so we did not do this for both years at the moment. But we can see some correlations between different categories we tested and the richness of the wildflower. So we can see that the BBCA's growing stage, the within-row spacing, and the Shannon index of the semi-natural habitats are positively correlated to species richness. And negatively influenced is it by the cover of the legume, the rotation, the ratio of the legume in the rotation, sowing depth, number of weeding measures, and also precipitation and aridity. And what I'd like to emphasize here as well is that we were looking at a correlation between the grain yield and different flora aspects as well. And we can see that there is a significant negative correlation between grain yield, cover of wildflower, and species richness. But looking at the Shannon index, which sort of also factors in the dominance of certain species, so the higher the Shannon index, the more balanced is the cover of the different species. We don't see a significant correlation, so meaning we can interpret that mostly dominant species are causing this trade-off between biodiversity and grain yield. Sorry, I'm always messing around the ones. So, preliminary conclusion, I'd like to point out here as well, as other partners were doing as well, we had an extraordinary hot weather this year, so this has led to an unbalanced data set in our data, and we also see that the year is a significant predictor of the species richness because we had some trial failures this year as well, and we see that hotter and drier weather leads to a negative effect on wildflower richness. Nevertheless, we are finding first leverage points in the management to increase wildflower. So if we want to increase associated biodiversity in lentil and chickpea fields, we should apply fewer or no weeding applications, we should apply monocropping, have a wider within-row spacing and a higher share of legumes in the rotation, and also be aware that the landscape context, if it's more diverse, it supports higher on-field biodiversity. We do have a trade-off between the species richness and the yield, but not between the yield and the diversity index by means of Shannon diversity. And it is important for further research to better understand the general richness potential that we are also serving farmers' fields and not only experimental sites that are highly controlled. And lastly, thanks to all the collaborating farmers in Italy, the consortium partners and external collaborators, and also to our biostatistician that was supporting us.
Thank you. Thank you. Are we ready for Nureddin? No. Okay, Nureddin, you can start.
Okay. You can start, Nureddin.
But I know I don't see my presentation. Thank you, thank you. Do you hear me?
Yes, yes.
Good morning all in the room. I am Noordin Yahya, and I work with Professor Beki at the Biotechnology of Rhizobia and Plant Breeding in University of Oran, Algeria. And so I think a great thing to Professor Federico for all work and effort in 3 years, in 3 last years in the Legum project. And I try to present you a summary of results of 3 years in this activity 5 and activity 3. For activity 5 and 3, there is 3 partners in our pipe. Then the table resume. The text, the table resume the deliverable for 2020 and 2021 years, 21, 22, and 23. So, for the first year, chickpea and lentil genotypes of partners were evaluated to irrigation regimes, 100 of evapotranspiration and 25% of evapotranspiration. For Algeria, there is 11 chickpea and 10 lentil genotypes were experimented in controlled conditions. And Spain, 19 chickpea and lentil genotypes were tested. For Tunisia, 11 chickpea and 12 lentil genotypes were experimented. So, as Federico has said at the beginning, in view of the results of the first year, the best drought-tolerant genotypes are retained for the following years. For the following years, Algeria has selected 6 chickpea with inoculated strains CC4 and the 3 lentil genpea in the inoculated with strains B119 selected at the biotechnological laboratory diriged by Professor Beki under 2 irrigated regime. And Spain, please, do you? There is no synchronization between the slide and my narrations. I saw that you are in the slide.
Sorry, Noordin, we are visualizing Spain results. You can go ahead with Spain.
Yes, Federico, what do you mean?
We are now on the Spain results. You have shown the results from Algeria, right? And now you can go ahead with the Spain.
Yes, Spain. For Spain, there is 3 experiments are carried out. In case of lentil field trial monoculture at the 3 seeds, Alba de Geto at Siena for lentil, and Carmona also for lentil station, experimental station. For the first, there is a monocrop in conventional management with 6 lentil variety with 2 mixed rhizobia strain JU2 and HA2. The second station, also 6 lentil varieties intercropping with barley, with 80-20 legume cereal, and barley in monocrop as control for the intercropping, other plant without fertilization. And the end, for chickpea at the Carmona CV, 10 genotypes, real advanced line and varieties. For main results for Spain 21-22, for the lentil, the best grain-yield lentil varieties come from Fuerteventura, Iceland. Lentil variety from Castilla-La Mancha did not perform well under these hydroclimatic conditions. The microcarpa variety from Fuerteventura, Leon, and Granada did show the highest levels, yields. And the microcarpa variety from Leon and Granada produced the highest straw material closer to 7,000 kilograms per hectare. In general, intercropping systems with barley do not negatively impair the grain or straw lentil yield. For chickpea, not find great differences in yield among both main treatments, and chickpea yields were similar after wheat or sunflower and sunflower. A great differences in yield among varieties, and the 3 outstanding with more than 100 were selected as real 33, real 92, and real 33. For Tunisia, 4 experiments carried at 3 seats: Saïda Manouba, Taxila Nabel, and Bushkir Nabel. For the first seed, there is intercropping potatoes, chickpea, rhizobia inoculation, and pea fertilization with management. For Taxila Nabels, lentil trails, genotype mixture inoculation, and with management. And the last, for Bushrik Nabel chickpea stations, also it's the same trial with lentil. The main result of Tunisia for this year, 21-22, that mean lead to select 2 tolerant genotypes for chickpea, Bushra and Rabha genotypes, and for lentil, 1 genotype was selected, Arabi Bio, that's named Arabi Bio. For Spain, in the year 22-23, the same schemes as in 21-22 are made with the 3 locations. And the main result for this year, due to very severe climatic condition of this year, which saw a very significant drought, the harvest could not be done at the experimental fields in both provinces, Sivi and Sion. In Algeria also, we have done an intercropping system with cereal anti-inoculation rhizobia strain that are selected with different partners, named B119 in Algeria and HA2 and GU2 in Spain, I think. The main result in Algeria, in the same cases of Spain, due to very severe climatic conditions of this year, which saw a very significant drought, the harvest could not be done at the experimental field in both seeds. The study of the barley and the lentil association in this experiment will be not possible since the barley crop has been completely destroyed by fungus and drought. we were enabled to do the inoculation experimental with different strains in these conditions. So, briefly, I present you activity that 3 partners also were implicated or involved in this activity. There is Algeria, Spain, and Tunisia. The table resumed the deliverable for 3 years, 2020, 21, 22, and 23. In view of the result of the first year, the best genotypes are retained the following year. Algeria 6 chickpea related with strain CC4 and 3 lentils genotypes were experimented with inoculated B119, and Spain 6 lentil genotypes were tested in inoculated strains GU2 and HA2. In 23, Spain and Algeria were tested a lot of genotypes of lentil and the chickpea, with different strain. Especially in Algeria, we have done 2 drought regimes, 75 and 25% of evapotranspirations. The main result in Algeria, as you saw in the figure, the result shows that all lentil genotypes positively respond to Rhizobia inoculans strains, whether in chickpea or lentil genotypes. For example, for lentil, Siri 2029 and TB-Moon genotypes associated with B119 Rhizobia strain showed the best result for root dry wild. And for chickpea, the genotype CD-Moon combined with CC4 Isola showed the higher result for dry root wild also. For Spain, lentil genotype versus Rhizobia leguminosa interaction, 4 lentil genotypes and 4 Rhizobia strains were tested. For the results, all lentil genotypes are positively responded to Rhizobia inoculants with symbiotic efficiency ranging 5100 to 500% over the uninoculated treatments. In 22-23, we have done the same experiments. 2 lentil genotypes displaying the contrast drought resilience and combined with 3 different used. Choice of these genotypes was made from samples of part of the produced in the laboratory in 22 of the project Leguminose. As you saw, the figure shows that experimental design, after 21 days from sowing, 3 individual reports per month and per repetitions are carried out for the best for a vegetative phase. Water deficit was imposed during the vegetative stage for 3 weeks and followed by re-watering period. Blue stars indicate plant sampling after 21 days of stress for different morphophysiological trait measures, and orange stars indicate harvested. Purple arrow heat indicate population spaced time with different strain Rhizobia. A group of morphophysiological traits were assessed. Morphological traits: the aerial part length, root length, shoot length, ratio root to length, lateral root ramification, number fresh wads of plant, and primary branch number are also calculated. For physiological traits, the total chlorophyll content was determined by using SPAD 5 0.02, the relative water content and dry matter were equalized. For yield compounded, the yield compounded number and weight of pod per plant, as well as the number and weight of seeds per pod and weight of 100 seeds were carried during physiological maturity after rewatering. Some comments about this result, these trials. The study showed that drought stress significantly reduced all traits in both genotypes in comparison with control. This reduce is significantly improved with the different association of rhizobia strain, particularly with A2 and GU2 strains. The parameters put the number of lateral roots can allow the identification of best genotypes with root characters superiors. These characters can help for a better recovery of nutrition and water from poor soil in semi-arid areas. It seems that the stability of the yield does not depend solely on the capacity of the plant.
Noureddin,
Yes.
please, two minutes.
Yes, okay, I finish. But also their ability to recover after stress. This ability to recover after stress is different in the two genotypes, depending on the type of stress associated with genotype. Thank you for all in the room, and thank you for your attention.
Okay.
Okay, thank you. I think we have to move on because we are too much late. And next speaker is also online. I hope the connection will be okay. Dulce, are you hear me? Can you hear me? Dulce?
Yes, I was
okay.
out with the
I will ask you to be very quick in your presentation because unfortunately we are late on the program.
Yeah. No,
okay.
only I want to say to all of you, if you can hear, listen me, or hear me, that the most important results that we have achieved here in Spain have been already present.
You have to share the screen.
How?
Yeah.
How?
Click on the arrow.
Click here.
So, briefly,
unfortunately, we are farmers. We don't know nothing about the computer.
Yeah, that's right.
Please move on.
Now you can see me well.
No, Dulce, you need to click on the arrow that goes up.
Arrow here.
The arrow button.
Arrow button. I did, and nothing come out. You can hear me at least?
Yeah, but we cannot see your screen.
Well, sorry. Well, the thing is that everything has been done, or things that we have done in Spain have been explained already. Mostly the experiment under field condition in two places in the north of Spain, where lentil is supposed to be a traditional crop, and also in the south, Carmona, where is an area not
Maybe, Dulce, why don't you send me your presentation by email, and then I do the same as I did for Noureddin. Send me by email the presentation.
Federico?
Yeah, do you hear me?
Yes. I only want to summarize what it has been done in Spain.
Okay, no problem. You can summarize.
Yeah, they have already talked about the Spanish trial and so on. One point is that I was saying that 2 localities were planted for lentils. In the north, Cuenca is a traditional area for lentils. And to our surprise, in the south, with less tradition, let's say none, or not so much tradition, we have obtained nice results in terms of yields. And in the case of chickpeas, you have seen we have evaluated 10 and bread of, let's say, new lines, and the behavior, agronomic behavior was quite good. On the other hand, I was to state that most of the best performer rhizobia strain for both lentils and chickpeas come from Spain. And also, as Ana Elena has stated, we can help Ana Elena in 2 years for her work on wild flora and so on and so on. And also this year, also she went to Cuenca. Pity, and she said this year have been really, really hard. So that's it. And we don't have results from the field experiment of this year, and we continue to explore the symbiotic properties of these unbred accession lines overall of lentils. Thanks. That's it. Thank to you. Thank all of you.
Okay. Thanks, Dulce. I think we can go with the next one. That is Christina. Christina von Hart.
Christina is the last one. I think there is an addendum to Christina talk at the end. I hope you both will stay in 20 minutes. Okay. Yeah. Okay. Malta is connected. I hope. Well, first, we will have the presentation by Malte Virguts, away from Leibniz University, and then I hope you will stay in 20 minutes anyway. Okay. Malte, can you hear me? Malte?
Yes, I can hear you. Okay. I hope you can hear me as well.
Can you share your screen?
I think you will have the presentation in Italy, and I will talk to you.
Malte, we cannot see your screen. Probably it's a problem, as our problem, not from...
I think the plan is to have the presentation by Christina, and I will do the commentary.
Okay. Yes. Okay. Okay. Please go on.
And I can...
Okay, wonderful. So I'm Malte from Germany, and I will present you the results from activity 8 on the cultural values provided by legumes. Next slide, please. So last year we performed a systematic literature analysis using Web of Science. We identified 159 records, of which 29 reports were included in the analysis after the two-step screening. And next slide, please. Most of the papers cover nutrition and diet and show moderate to poor adherence to the Mediterranean diet. The papers also indicate that too few legumes are consumed across all ages in Spain and Italy. The papers also indicate, so for the cultural ecosystem services, one paper showed that legumes are often mentioned in traditional Spanish songs, and Mrs. Pia Giovanni, maybe she's with you today, pointed out that legumes historically assumed great relevance in the religious and social events of the Apulia region in southern Italy. Next slide, please. Also last year, stakeholder interviews were performed in Italy in the area of Castelluccio di Norcia, which is a PGI region with a famous lentil-shaped agricultural landscape. Next slide, please. 22 local stakeholders with different backgrounds from various sectors were interviewed. For example, among the interviewees were representatives from agriculture, nature conservation, and tourism. Next slide, please. The perception of the ecosystem services shows that cultural ecosystem services, displayed here in purple, rank among the highest scoring ecosystem services. Especially the sense of place, the aesthetic and the recreational quality of this lentil-shaped landscape are highly valued by the locals. Next slide, please. This year we prepared an in-depth interview to be conducted in Italy and Spain. We created an 8-page questionnaire with a qualitative and a quantitative part. We contacted 48 suitable interview partners in regions with a high proportion of lentils or chickpeas in agricultural production. We contacted tourist associations, agriculture and legume associations, as well as individuals with expertise in the field. We received only a few responses, and due to the low interest in participating, we were only able to conduct 2 interviews. Next slide, please. Yeah. Even though the significance of 2 interviews is low, there were many commonalities. For example, legumes are firstly associated with gastronomic aspects. Legumes are still important for the local culture. There are many traditional dishes with legumes that still are cooked and served in local restaurants. Legumes contribute to the local identity. And both interviewees had no knowledge about religious or spiritual meanings of legumes, but the Italian interviewee had 3 local festivities in mind in which legumes are central or play an important role. Next slide, please. And currently we are still working on an in-depth literature analysis of primary sources. We identified poems, recipes, and folklore and fairy tales as suitable sources to gather further insights into cultural values provided by legumes. For the poems, we created a web scraper in Python that automatically collects the data from the suitable websites. So far, we have collected over 8,000 Spanish poems, and the Italian poems will be collected next. For the recipes, we have identified suitable cookbooks and websites. The collection of individual recipes is about to start very soon. And for the folklore and fairy tales, we so far identified 1 book and 5 suitable websites in which legumes were mentioned in 8 individual stories. But the collection of suitable sources is still going on. And all these primary sources will be analyzed with regard to the mention of legumes and the context in which they are mentioned. So for the lessons learned, we learned that most scientific publications deal with the legumes in nutrition, and only a few cover the cultural values. Interviews especially can give great insight into the cultural values provided by legumes and the locals' perception of legumes. Our first implementation of the web scraping is very promising, and we think that the further analysis of the primary sources will yield great results. And last but not least, most important, the knowledge about the cultural significance of legumes is important for the promotion and acceptance. Thank you very much.
No, that was it. I think Federico is doing, yeah. stay here for the discussion, but we can do later.
I think we can go ahead with the discussion quickly if there are any questions, right? If there are any questions from the audience, we can do the same as we did this morning. Open questions.
Maybe one afterthought about the presentation we just saw and heard. This is a kind of framework for promoting, of course, legumes. We all know that legumes are very important in the diet of people in order to compensate if we reduce meat consumption. On the other hand, legumes from the time of the Bible have an interesting cultural meaning, also religious meaning. You all remember the legume dishes which played a role in the Bible.
Let's hope they will provide some peace. Now we need some.
Yes. Maybe we should send some legumes over there. But this is, so if we want to promote legumes, it is not only by scientific argumentation, we think, but also by appealing or by promoting legumes in telling a story about legumes. And this is what we look for, to have some empirical evidence of what is going on in the regions, whether there are some basics to which we can link that people still know from their parents or whatever, and develop something like, maybe it's even a business case, which what we are looking for.
In this presentation, there are only lentil and chickpea. Consider also beans that are not local, at least in the ancient time, they are not local. Beans are considered also, because there is a lot of different beans cultivation in Italy, at least. And
in the literature analysis, we considered edible. I mean, legumes suitable for human diet, so beans are also part of that. But on the other hand, we have only lentils and chickpeas for the biodiversity, which is a very nice side effect, as we see, and even may not interfere very much with the yield, as Annalena pointed out. If you look really at the biodiversity, at the species richness, and do not. Also, the species richness is suffering if you have some very dominant weeds, such as Gallium aparine or something like that, which is overgrowing not only the crop but also the wild species. So there may be something like a synergy even, if you apply certain management measures. And what you can also deduce from her findings is how should agri-environmental measures be programmed. We had it this morning that there is not enough scientific evidence in the programming of the official agri-environmental schemes, but also, so this is something which can help farm advisors and also success-oriented remuneration of such services. Because the farmer knows what to do, and then the success can be remunerated.
Yeah, Francesco, can come here.
Yeah, the health, it's true. The health benefit we didn't address. Yeah.
This I think is a really important
yes.
point.
Thank you. Yeah, thank you. We know about that, but
Because we wanted to say only the good things about legume, but it's true. We should also focus on some negative aspects, and in order to reduce the amount of that, yeah, it's true, should be done much more. In legume, we are focusing more on the good things, like flavonoids, polyphenol. But in the project, there is also a part that is related with anti-nutrients that we have not done yet. That will be very, very important to do it. And I really hope that the partner that we have, the CNR, should do, in addition to this, also analysis of these anti-nutrients.
Yeah, the anti-nutrients is one thing, but you also have to see the trade-off between reducing your meat consumption, which contributes to your health, and then in consequence eating more pulses, and on the other hand, the anti-nutrients. That's true. Yeah, or the toxicity. Yes.
Anti-nutrients are bad for us, but they are not bad for the legumes. They survive in their environment because of
Yeah, yeah, yeah.
So let's be careful.
Yeah.
It's true.
So you might also imply that they had to reduce pesticides, for instance. Yeah, yeah, yeah. Aha. Yeah, yeah. Interesting.
There are any other questions or points that you want to address? No? No question? Okay. Okay, so I think we can... Oh, yeah. Francesco? Yes, Francesco Pini can make the conclusions. And after that, don't forget to come here to make the picture, okay? Because the picture is very important, so please do not escape, come here, because we need to do the picture.
Good evening. So I'm here for the concluding remarks, but I will be very short, I promise. So first, I want to thank all the speakers that have been here today with their beautiful presentation, sharing their results. And for these concluding remarks, I will go back to the roots, so at the beginning of this day, and at the beginning also of what we do. Sometimes we are working on this field, if we compare what we do from a scientific point of view and what we see on the market, there is a big discrepancy. We still use old bioinoculants. We are going to go back to old farming system, as the directors today pointed out. We are going back to the past. We are watching to the past. So we are just reinventing the wheel. And sometimes I wonder myself if it's so. I share a couple of slides with Marco, but I've done my PhD with him, so I think it's normal. So two major points in the past century have been the Haber-Bosch process and the Green Revolution that has revolutionized our way to do agriculture. And if we see how increased the population in the past century, we see how these two processes, these two ideas completely changed our world. Anyway, everything comes back with a price, and as Marco has shown, this is the social cost of fertilizer, climate change, air pollution, surface water contamination, groundwater contamination. So we have many problems that are related to our actual way to do agriculture. A few years ago, the team of Professor Rockstrom proposed what are planet boundaries. These boundaries represent which are the major problem for our world, for the world health status. Here we see that we have different colors, green, yellow, red. The planet boundary is in between the green and the yellow area. The green spot are the safe place where we can still have a condition that it's the Anthropocene, that it's a word in the environmental safe condition that is good for us. But if we see all these different fields, for some of them we're already in the red zone, in a danger zone, especially for nitrogen and phosphorus biochemical flows. And if we see how agriculture is related to these different planet boundaries, it's related in every different aspect, from loss of biodiversity, change of land use, and especially in what all the biochemical flows, in nitrogen and phosphorus. So coming back to the question I have for myself as well, do we need rocket science for bioinoculants and farming crops? Indeed, yes. As we saw today, even if we don't see something that is already on the market, we are now in the right direction. In the past 40, 50 years, we have accumulated a lot of knowledge about everything that at the molecular level on how Rhizobia legumous plants works. And now we have the knowledge and the methodologies with the development of all the omics, genomics, transcriptomics, to go in the field. If in the past 40 years, most of the research has been closed in our lab, now we have the resources and the knowledge to move on the field and test what is effective to have a sustainable agriculture. So another question that we may have is, are we ready for a next sustainable green revolution? Not yet, probably, but we are in the right direction. We just have to move faster because the clock is ticking for our planet, and we saw every day checking how the climate change is going faster and faster. Anyway, so we have to try new farming system, as is pointed out this morning, and also this afternoon, new bioinoculants that should be effective, new way to select plants and to increase biodiversity. Everything that we saw today, it's in that direction, and we are just move faster. Also, other ways to recycle, as we see in the presentation of group of Professor Melen Buccioni, about recycle everything that is considered still now a waste. Anyway, it's not just about science, what we saw today, it's also about connection. Here, I enjoy myself to color all the different countries that are involved in the 4 projects we saw today. And we see how all the countries in the Mediterranean area, most of them are involved in a project related to the leguminous plants and sustainable agriculture. So it's not just a question of science, but it's a question of sharing, to be more in contact also with the stakeholders, farmers, and understand what are their problems and how we can solve it. So it's just we need more connection between science and farmers, and politics should be in the middle between us to facilitate research and connection with the problem that agriculture has nowadays. So I finished, I promise. I was very, very short, and I thank you for your attention.
Thank you, Francesco, for summarizing our work in a very nice way. So thank you for the audience, from the people that... Please conclude.
Okay. Yes. As Carlo said, I would like to thank everybody for the participation, and above all, thanks Carlo for the great effort that he spent for the organization of this event. And personally, I'm very happy to work with legumes, and I hope that in legumes we can develop future projects, because I think even if they have some weakness, and it's very hard to work with them in control condition because of they do symbiosis. As we find, and as we realize today, they are very, very important crop for healthy perspective, but also for sustainability. So let's continue to work on that, and let's hope that this event will help us to interact more and have an opportunity to develop future projects. Thanks also for my colleague Alessio that also participated actively on this event, and thanks also to our mentor Marco for everything, for the support. And let's hope that we can have another event together in the future with another exciting project. So please come everybody for the picture, because as I said, it's very important deliverable. So, and for everybody that wants to come to this night, please, the meeting will be at 7:30 in the front of this place, the rectorate. So 7:30, around 7:30 we meet here, then we walk to the restaurant that is close to the train station.